I was speaking with Dr. Reinhardt. He said that a lot of people have approached you about like how this can impact, like, help against neurodegeneration, and just a lot. What type of greater impact can this have on the medical community, do you think?
I do not know anything specific about neurodegeneration, but you probably know about the phosphoprotein. So the phosphoprotein in mammalian cells in humans is a very large number of phosphoproteins that contain, among others, phosphoserine. And these proteins are very, very important because they are, as you said here, on-off switches for many processes. So clearly they're important for neurophysiology, but for many, many other things. But to study them has been exceedingly difficult because there are so many of these kinases, enzymes, and phosphatases, and they're not pure, and they cannot be synthesized, because unlike what we have done, phosphoserine is not put into the protein per se. It's serine on a protein that gets phosphorylated. And some other protein comes, phosphatase takes that serine off again, and then it's active or inactive. But the relationship of what is the substrate and what's the enzyme is unclear because it's all a mixture. So with this then, at least the phosphoserine containing proteins can be synthesized in E. coli. And then you can go and study this in detail. So lots of people have asked already, could they have the various clones and bacterial strains so they can apply to their research. So it will be a tremendous research tool for many, many people studying human physiological reactions.
Do you know of any other research that's already being built off of your research?
I mean, people have come and said, I'm working on, and this was the first one, on a plant signal transduction protein, and it was a plant hormone, and we can now synthesize it, and we can now find out how is it really used, and what degrades it. Many people who have phosphoproteins, phosphoserine proteins, human proteins, can now for the first time say, this is the enzyme, now I can study it, how active is it when it's changed, and so on. And the method also allows to make, as a matter of fact, phosphoserine containing proteins that are unlike the human ones, because the human ones, it says, let's say, my phosphoserine is in position, let's say 200 of the protein. I can now put it in position 198 and see what it will do. In nature, this cannot be done, because there are phosphorylation sequences that say it goes in here, but I can't put it one away from there. So it cannot be done. So you can actually get synthetic proteins and see what they do. Do they inhibit? Do they do something different?